Alright, so this video will be talking about X inactivation and associated Barr bodies. So we know that females, they're going to have two X chromosomes, so XX, men are going to have XY. So we're going to be focusing on the XX in females right now. So what happens is in X activation, we're essentially shutting down one of the two X chromosomes present in the female. So this process is indiscriminate in humans at least. So either X chromosome has an equal probability of getting shut down and inactivated, hence X inactivation. So once this X chromosome is going to be inactivated, it's going to be confined to a very heterochromatic state. So we know from previous videos when the chromatid's super heterochromatic, it's going to be really tight and coiled. That coiled nature of the heterochromatin and chromatin is going to mean those transcription factors and furthermore, RNA polymerase isn't going to be able to bind with the DNA and create that mRNA, which is necessary for gene expression, right? So basically, we're shutting down one of the chromosomes, and that chromosome is going to be condensed so much, it's going to be called a Barr body. And it's going to be super, super tight and inaccessible. So this Barr body, which is the product of this X inactivation, this was discovered by a British scientist named Elizabeth Lyon, and it's often this process of inactivating an X chromosome and reducing it to a very heterochromatic state is known as lyonization after her. So the genetics behind this phenomenon explains why Turner syndrome and triple X females can survive. And so Turner syndrome, that's going to be 45 with just the X, right? And then because only one chromosome is needed, the remaining X chromosomes can be crumpled into Barr bodies. So Turner syndrome, 45X, there's no Y, and there's only one X. But while there's going to be some developmental issues and potentially muscular issues, in general, the person is going to be somewhat healthy because they only need one X chromosome for survival. If they had more, they would just be crumpled into Barr bodies. And then the same goes for the triple X female. Only one of those X chromosomes is going to be used, so the other two are going to be crumpled up and made to be super heterochromatic and inaccessible, so they can't be transcribed. So the classic example for this X inactivation in Barr bodies is going to be calico cats. So calico cats are only going to be females, and this is because you need the XX nature, right, for that Barr body. So when you see orange spots, you want to think that's the result of the black allele being crumpled up into that Barr body while the X is active, or while the orange is active, excuse me. And then when you see black patches in the cat, you're going to think the black is going to be active while the orange is going to be all crumpled up. All right. So I hope that explained X inactivation and Barr bodies, and thank you.